I have a love for biology and living organisms, for human health and disease, and for the environment and the world around us. At the start of AP Research, I didn't know which topic I wanted to pursue, but I knew that these were my passions, and that I somehow wanted to connect each of them. I began my research on the topic of climate change, as this is such a huge topic with so many potential areas to investigate. I learned that this is an area that has received a lot of attention in recent years, and as a result, a variety of studies have been conducted. Essentially, these studies conclude that on a general level, increasing temperature actually increases the fitness of organisms. I decided to research how temperature could impact an important disease vector, such as the mosquito. From here, I looked into mosquitoes and the diseases that they transmit, such as malaria, dengue fever, and West Nile virus, to name a few. Because mosquitoes are responsible for hundreds of thousands of deaths each year, it is critical to consider the effect that climate change will have on their fitness level as geographic regions begin to shift. As stated, the current hypothesis of scholars in this field is that as temperatures increase, insect species will generally become more apt to survive and reproduce. This is determined by emergence rates and fertility of mosquitoes, meaning how many larvae survive to reach adulthood, and on average, how many eggs do they lay. However, studies pertaining to mosquitoes focus primarily on species living at or near the equator, where climate change is expected to have a much more significant impact than more temperate regions. The gap that I then identified in research is a general lack of empirical data on a wider range of mosquito species living outside of the tropics. I selected the genus Culex. Ultimately, my research question became, what effect, if any, will the increasing average temperatures of the globe have on the emergence rates and fertility of Culex mosquitoes at more northern latitudes? To answer this question, I decided to use the experimental method. The particular method used was a combination of several other experiments used in similar studies. I began the experiment by setting up 3 tanks to serve as microenvironments. Each tank was to be kept at a different temperature. Tank 1 was kept between 23 and 24 degrees. Tank 2 was kept between 25 and 26 degrees. And Tank 3 was kept between 27 and 28 degrees Celsius. The orange region on the map averaged 75 degrees Fahrenheit, which is approximately 23.8 degrees Celsius. This temperature ultimately became my control group. The experimental group temperatures were obtained from estimated increases for the years 2030 and 2090. To heat the tanks, heat lamps were used. 150 Culex larvae were added to each tank and approximately 1,000 milliliters of water. After setup, 4 weeks were given to allow the larvae to reach adulthood. The adults were then sustained on 10% sucrose solution until the end of the 4-week period. This photo demonstrates my setup. After the 4-week period, blood was introduced to induce reproduction. A 24-hour time period was given to allow females to feed, after which the blood was removed. The blood used in the experiment was prepared by grinding up a 4-ounce cow liver in 100 milliliters of water. And yes, it was as gross as it sounds. From here, 100 milliliters of beaker were filled with the blood. Parafilm was then stretched over the top to replicate skin, and the beakers were placed upside down on top of each tank to allow for feeding. An additional 24-hour period was given for oviposition, meaning laying eggs. Following this period, the filter paper substrate was removed from each tank, dried, and examined underneath a microscope for eggs. Throughout this experiment, temperatures were recorded twice daily, in the morning and in the evening. All 3 tanks had average temperatures within the established ranges prior to the experiment. Also recorded on each day was the number of adults present in each tank. Tank 1 had a peak of 9 adults, Tank 2 had a peak of 2 adults, and Tank 3 had a peak of 4 adults. Unfortunately, no eggs were laid in any of the tanks at the end of the experiment. Now you may be thinking, as was I, that this means the experiment failed. However, I was advised to run statistical analysis on the peak number of adults in each tank anyway. A 2-sample t-test was conducted on the peak emergence numbers of adults to verify their significance. Essentially, the results of these tests suggest that increasing temperature actually decreases emergence rates, and therefore decreases the fitness of the mosquitoes. One study, conducted by Athene Costa, found similar results on a different mosquito species at more dry climates. However, when humidity was increased, emergence also increased despite temperature. This study both helps to validate the results of my study and suggests that humidity is a more important factor than temperature in determining emergence rates. My study was conducted in Colorado, obviously, which historically has very low humidity levels. Because of this, emergence rates may have been influenced by the drier climate, and in fact, this could account for the decrease in fitness seen from increasing temperature. The results of my study yield two conclusions. First, increasing temperature at lower humidity actually decreases emergence rates of Culex mosquitoes. And second, temperature may play less of a role in impacting future ranges of mosquitoes than previously thought. And in fact, humidity may be a more significant factor than temperature. So the question then becomes, why should we care? What do these results really mean for our future? Well, ultimately, the conclusions reached from the study are in fact contradictory to the current general hypothesis, in that temperature was expected to have a direct relationship with mosquito fitness, but instead had an inverse relationship. While my results do not necessarily disprove this hypothesis, they do call into question our true understanding of the world around us. Indeed, these results suggest that this topic requires further research into the effects of humidity on mosquito fitness as opposed to just temperature. Such research could have drastic implications as to the potential prevention measures and protocols seen for fighting mosquito-borne diseases as climates begin to change. Thank you.